All right, so now we're bitten by chikungunya. We are into the first week of this illness. We passed 4 days, and our poor patient cannot get out of bed. He is bent and broken, as the word describes. And this is a picture from Google Images of a patient in the Caribbean with chikungunya who is really incapacitated. As I pointed out, not only is there expansion of immunocytes, but there's inflammatory markers right now. And we now require different assets to overcome this danger signal. So here we have what we are recruiting. At 30,000 feet, we need T cells to engage in cell-mediated immunity. We have T helper cells, these CD4 cells, which have amazing complexity, which will lead to activation of further other recruiting cells of polys and monocytes, eosinophils and mast cells. And then we'll have CD8 cells, which are cytotoxic cells. And at the same time, you're going to hear later about recruiting B cells to make plasma cells and antibody and generate memory. So T cells are the predominant cell in the peripheral blood. As you all recall, 3/4 of our lymphocytes are T cells. They derive through the thymus. They are recognized by a variety of cell surface proteins, including the clonotypically expressed T cell receptor and its attendant structural proteins. Here we have a picture of the T cell receptor. 95% of our cells have the alpha beta version. There's a small percentage that are gamma delta. And here we have the non-covalently linked accessory molecules that co-migrate, co-cap, and actually provide the signal transduction material. This is known as the CD3 complex. We know from the Nobel Prize winning work of Tanagawa and others that this T cell repertoire, which contains 10 to the 10th, 10 to the 11th, some huge number, is generated through this post-germline somatic mutation process. So it is a very dicey thing. We are generating almost an infinite number of T cell receptors, and we need mechanisms to make sure that they don't react with self, and Dr. Rigby is going to talk about that. The T cell receptor itself has a very fine area which it binds to its cognate antigen. Most of it is fairly stable molecularly, but there is this complementary determinant region that has a very high frequency of molecular changes that allows it to bind many different antigens. It will only recognize an antigen when it is presented with endogenous, your own HLA molecules. You can't recognize the same antigen presented by an unrelated person's HLA molecule. So this is a safety mechanism that keeps control of this. Antigen presentation, as we'll talk about through a number of times, can be presented in the context of MHC class II, HLA-D family molecules. These present to CD4 cells, and HLA class I molecules, A, B, and C, that present to CD8 cells. In a reductionist view, the class I molecules, A, B, and C, will present antigens that are derived from within the cell. So viruses that's residing within the cell and infects the cell can then be degraded through proteasomal pathways and will ultimately surface on the cell. And so when you get the influenza, that CD8 cell can come and knock it off. On the other hand, many of our infections come to us from outside the cell, bacterium, mycobacterium, and then they invade these extracellular antigens. They're processed through the MHC class II pathway and ultimately resurface looking for their cognate CD4 cells. Now, if that is antigen presentation in a minute, the challenge now is to have appropriately processed and presented antigen. Now the T cell must find that cell presenting the antigen and must receive signals to allow it to become activated, proliferate, and go do its thing. So how does that happen? Well, there are several things that are happening all at the same time. We now have canonically recognized that it takes more than antigen recognition to activate a T cell. I mean, if it was that simple, every time a T cell would see an antigen on the surface of your cell, whether it's lit by a bacterium, a virus, some type of viral stimuli, and it bound to it, we would be killing cells all the time. We have to protect this. So it requires a fail-safe, and that is a second signal that we'll talk about. And in the absence of this second signal, the T cell will not become activated. There are a variety of second signals that can participate in T cell activation. The one that we know the most about at the present time is CD28 binding to a family of molecules known as CD86. This is important because now we target this for autoimmunity, we target this in cancer. You'll hear a lot about this in the next day. CD28 is borne by the vast majority of CD4 cells, about half the CD8 population, and it binds to this family of molecules, the CD86, on the antigen-presenting cell. If you don't get the binding of this second signal, the T cell not only does not become activated, but it may undergo apoptosis and die or become anergic, meaning that it cannot be turned on even by direct stimuli. So it's programmed not to respond. An activated T cell will release the cytokine interleukin-2, which is known as T cell growth factor. This is showing that the two signals are required for interleukin-2 secretion. T cell growth factor allows cells to proliferate and go on to their effector function. Now, another term that you should recognize right now is the immunologic synapse. So, you know, it is not just this T cell here that is seeing this antigen on an antigen-presenting cell. It's not just like these two molecules coming together. There are literally hundreds of molecules that will migrate in this fluid membrane of the T cell in this process that descriptively was called patching and capping. We now have more molecular terms of talking about lipid rafts that allow these molecules to congregate and to move. But these include adhesion molecules, which increase the binding affinity between these cells and allow them to be oriented in a way that the cytokines can be directed in an appropriate manner to be most biologically active. This is just to give us the flavor that here we have the T cell receptor, here we have the cognate antigen, here we have the second signal CD80 and 86, but there are other co-stimulatory molecules, such as inducible co-stimulatory molecule known as ICOS, CD40 ligand, and adhesion molecules. And I put these up here for this recent Nature Reviews in Nephrology to show that there are already molecules in clinical development that target each and every one of these pathways, looking at them in terms of their capacity to suppress transplant rejection. So we have to be aware of this and recognize that it's CD80, CD28. This is not the only show in town. So once this effectively does occur and a T cell is activated, what happens? Well, here's the infection. Here's chikungunya. Here is what we see in terms of CD4 cells. Within a week, we have multiple log phases of a single cell replicating, and also recruiting CD8 cells, these cytotoxic cells, which actually can kill virus harboring cells. But within a week or so, there must be homeostasis, or every time you get the flu or a rhinovirus, you develop a lymphoma. And you have to have signals that shut this thing down. It is vitally, vitally important. And so by a couple of weeks after a successfully dealt with infection, we are regressing, and we maintain a memory response. Certain infections like hepatitis C, which replicate several times a day for 40 years, violate this, and we'll come to the consequences of that in a while. So everything has a good and a bad side, the yin and the yang, and here is the break. So if signal 1 and signal 2 accelerate this, the break is mediated by a family of molecules, the most prominent of which, because it was elucidated first and has been exploited immunotherapeutically, is CTLA-4. CTLA-4 is similar in structure to CD28 in as much as it will bind with even greater affinity to the CD80-86 family. It is not nascently expressed on most T cells except for T regulatory cells. And once the T cell is activated, it starts to sprout and grow CTLA-4. Because it's more avid, it then kind of kicks off CD28, and then once it binds to CD80 or 8086, it sends a negative or a control signal to the T cell to stop replicating, to slow down. So it is a break. Here is basically showing that once a T cell has expressed a lot of CTLA-4, it will co-opt the second signal, and instead of a driving force, it will be a negative force. Again, if it were only that simple and there was a starter and a break, it would be easy. But this is a more expanded view of co-stimulation and co-inhibition. And here are numerous other ligand-ligand pairs that can augment different cell populations, different cells under different situations, and families of breaks. We will focus on CTLA-4 and PD-1, but be well aware, and I'll show you at the end, there are developmental therapeutic molecules for a variety of these. Now tomorrow you're going to hear an expanded talk about tumor biology, but this is just the pictures. So for generations, it has been the dream of oncologists to be able to capture a healthy immune response to overcome cancer. In certain ways, patients with immunodeficiencies have increased rates of cancer, so it's a failure of integrated immunity. About a decade ago, this experiment was published in the New England Journal and has influenced the way we think about immunotherapeutics since that time. This is the T. Gennaro company, TGN 1412. This is a very famous paper because it showed us how poorly we were doing these clinical trials. This company came up with a molecule that preferentially targeted T regulatory cells. You'll hear more about these a little bit. So in other words, this was going to take the brake off the immune system in a very, very selective way and allow an outgrowth of anti-tumoral T cell responses to go. So it was very good in theory. Unfortunately, the experiments were only done in preclinical models. This was not, there was no human homologue of this, so the preclinical work was very, very inadequate. And this is the hands of one of the first 6 subjects that were given this first-in-man phase 1 trial that experienced profound immune activation, cytokine storm. And the lessons learned, in addition to the fact that we had not paid enough attention to the lack of good preclinical modeling, and this should have been done in a safer way. Those of you out here that nod and know this, know that each of those 6 subjects were given this drug at the same time. They're all lined up. So not only did they have one person experience this, there were 6 people in cytokine storm. And now, any first-in-man trial, you give it to one patient, you wait many days past its PK, PD, and then you do it again. So a lot of lessons were learned. So ill-fated. So today, I'll introduce the notion of immune checkpoints because we've already talked about CTLA-4. 2 immune checkpoints are this first pathway, which is basic to T-cell deactivation, CTLA-4, CD28, and then a second pathway that known as programmed cell death, or PD-1 pathway, which occurs more in the periphery and occurs somewhat later. These are not bad ligand pairs. These are not negative to our immunologic health. They're actually vital to our health. And in fact, this PD-1 pathway I was talking this morning, this is actually quite physiologically important, and maybe Dr. Silverman might talk about this in terms of the generation of a robust B-cell response, because PD-1 is expressed on T follicular helper cells, which are driving this differentiation. So when we target this, there may be prices to be paid down the line. Again, a highly complex array, and I'll leave this to tomorrow to our talk in oncology. But there are 2 drugs that are now been approved as targeting these checkpoints. Here is ipilimumab, which targets CTLA-4, which has been, have some very remarkable data in melanoma. And what is the downside? Well, if you're targeting the brake, take your foot off the brake, you're going to get inflammation. And there's a very interesting array of adverse events that have been seen. This is a very serious condition, so our risk aversion is modest, but prostatitis, autoimmune skin disease, autoimmune central nervous system disease, this is now part of the new wave of toxicity. So here's the patient with the cold. Gets cold. Within 10 days, 1 T cell has become 1 million T cells. Within a week or so after that, it is contracting. 95% of these cells die. You're going to make a little bit of immunologic memory. Those memory cells will self-renew over a long period of time. But if for some reason you can't get rid of the pathogen, you have hepatitis C, HIV, hepatitis B, or a cancer, can't get rid of it. So now those cells can't contract in the same physiologic manner. And there is a term that you should recognize known as immunologic exhaustion. I think it tells the whole story. This is not an anergic cell. This is not a cell that is programmed not to respond. These are cells that are just losing it. And they lose the capacity to make IL-2. They lose their capacity to be cytotoxic. And they lose their capacity to generate gamma interferon and other signatures that have been looked at. So an exhausted cell. So we see them in chronic antigenic stimulation. So there's 2 situations where these are seen. One is in infection, and the other is in cancer. The infection ones are the organisms I've told you about, and you'll hear more detail about cancer. So one of the main arbiters of immunologic exhaustion is the aberrant expression of these PD PD-L ligand pair on the surface of these cells, so you can phenotypically identify them. These exhausted cells have an expanded phenotype, so it's not just PD-1. As again, if it were that simple, it'd be easy, but there are multiple other phenotypic markers in these exhausted cells. And these are being exploited now in early clinical trials, markers such as log3, TIM3, et cetera. These are quite different from anergic cells, not only in their cell surface, but in their transcription profiling. So it's an important thing. So these are just a couple articles that I expect you'll see more detail on. The first PD-1 molecule to be approved for small cell, I heard this just this morning, has been recently approved. And you know, I love these oncology articles because you only have to show a picture. And it's pretty great stuff. You know, in rheumatoid arthritis, you got the DAS28, CDI, SDI. Tumor goes away, that's a good thing. And we'll hear more details tomorrow morning. But I do believe, and I've heard this from my oncology colleagues, is that, you know, melanoma is one thing, but that's an immunologically sensitive tumor, and I could kind of get this. But small cell cancer of the lung, that's hardcore stuff. So seeing great responses in these patients has been interesting. And they are actually been used in combined therapy, and this has been shown recently in the New England Journal, as has been remarkable success, even in highly refractory Hodgkin's lymphoma, and there have been biomarkers generated to show this. So I wanted to just end with a kind of a summary of what I've attempted to do. So I hope, but at the end of this first hour and a half here, you have a much better overview, that 30,000-foot view of the integrated immune system, that you can recognize the key players in innate immunity and at least identify their primary functions. I hope that you recognize that there is a seamless and logical connection between innate and adaptive immunity, and that these are all vital to immunologic homeostasis.